Good day, everyone. Ladies and gentlemen, you are welcome to another lecture video on enzymes. So today we are going to look at enzymes, their properties, and what they actually do in the biological system. So enzymes, they are proteins that act as biological catalysts. They catalyze reactions in the biological system, and they are proteinous in nature. So there are other enzymes like the ribozymes that are of nucleic acid origin, but majority of enzymes that are used for catalyzing one reaction or another in the body system, they are protein. They actually carry out the process of speeding up the rate of chemical reaction, and without them being actually consumed in the process. So they will catalyze the reaction. Let's say you have a reactant A plus reactant B to form a product C. So the enzyme will facilitate the formation of that product C from reactant A and B, but without actually being consumed or depleted during the reaction process. So you have a substrate A and B, okay? Now, this substrate A and B, they are going to be combined or joined together by this enzyme, and the enzyme actually has an active site where there are some specific amino acids that are responsible for the activity of that enzyme, and that active site is where the catalysis occurs. Okay, you remember we discussed about amino acids that are found in protein, 20 common amino acids with all their different side chain and their actually properties. So the properties of those amino acids, they help in the catalysis of enzymes reaction, those amino acids that are found in the active site. So in the enzyme, you have a region that the substrates will come and bind, and then the catalysis will occur. But before that, when the substrate A and B binds to the active site of the enzyme, they have a shape that can fit into the active site, and they form what we call the enzyme substrate complex. Then the catalysis will occur, and you have your products C and D, with now the enzyme being intact. So the active site of an enzyme is where the substrate binds, and substrates are the reactants in the chemical reactions requiring enzymes. So enzymes are specific. There is a specificity that each enzyme actually is guided upon by what we call lock and key model or an induced fit model. So for the lock and key model, it is the oldest model that enzyme binding site was an exact match for the substrate. So it means just like your lock and key, your key fits into your lock exactly and perfectly. So the binding site of the enzyme is like the lock, okay? The binding site, the lock, and your substrates are the what? The key. So they form, they now interact with the enzyme. They are the active site to form this complex, and then the products is released, or the products are released by the enzyme, and you have the intact enzyme. While for the induced fit model, it is a model that explains that enzyme binds most tightly to an intermediate form of the substrate or the product. So this is a model that is more compatible with reversible reactions that are catalyzed by one enzyme. So you have some reaction that is forward reaction, and you have another backward reaction. It's a reversible reaction that the reactant can be used by the enzyme to form the product. So also the product can be converted back to the reactant by the same enzyme. So this model actually is compatible with such reaction, where the enzyme and the substrate forms the intermediate, which is the enzyme's substrate product, and then you have your product and the enzyme back. Then that same product can bind to the enzyme to form the intermediate and as well the reactant and the enzyme. So in terms of naming of the enzymes, enzymes usually end with ASE, okay, and typically their name indicates their function or the substrate they bind. So important examples include, you have kinase. Kinase, they add phosphate to the molecules, and you have the phosphatase, which remove phosphates from a molecule. You have the hydrogenase that removes hydrogens from molecule. Hydrolase, they add water molecule. Synthase, they serve as dehydration and the synthesis reaction. And you have carbonic anhydrase, which remove water from carbonic acid. Amylase, digest starts, they digest starch. And you have lipase that digests lipids, and protease digests proteins. Okay. So the enzyme specificity, it's referring to the ability of enzyme to catalyze only a certain type of reaction. Some are very specific, such as the glucokinase, and it catalyzes reaction where glucose and inorganic phosphate are actually acted upon by glucokinase to form glucose 6-phosphate. And other enzymes, they have broader specificity. For example, you have peptidases that break all peptide bonds, and you have the time and the amount of enzyme in the body can actually vary depending on the function and the role of that enzymes in the system. For example, you have alcohol dehydrogenase, which removes hydrogens from alcohol and thereby reducing its toxic effects. So now this is a simple question: is everyone's ability to handle alcohol the same? I want you to ponder on this question. Is everyone's ability to handle alcohol the same? So enzyme activation, some enzymes exist in an inactive state, and they need to be what? They need to be activated. Okay? So there's what we call, there are different types of activation, and we will look at some few, and we have the proteolytic activation, okay, whereby you have pepsinogen that is inactive and gets converted to the active form, which is the pepsin. And pepsinogen is longer in terms of the chain, and you have a shorter form of it as the pepsin. So other examples include fibrinogen. When it's activated, what does it become? Thrombinogen, what does it become? And when angiotensinogen is activated, it becomes, what does it become? So these are some actually few exercises that I want you to look at and now tell us the correct answer. So what is the active form of fibrinogen, thrombinogen, and angiotensinogen? So enzyme activation can occur via cofactors, and these cofactors are inorganic components or inorganic molecules that are required for substrate binding at the active site. Example, we have zinc, divalent cations, we have zinc, calcium, magnesium, or copper 2 ion, and we have enzymes that are requiring, as an example, that are requiring these divalent cations. We have carbonic anhydrase and alcohol dehydrogenase, which requires zinc ion, and cytochrome oxidase, which requires copper 2 ion. We have monovalent potassium and divalent magnesium that is required by pyruvate phosphokinase. So okay, so this is a chart that is explaining how minerals have been used up in the soil. Okay, you remember this the units. Look at the x and y axis, and the x axis is in years. Okay, from 1910 to 2006. And also the y-axis is in parts per million. That's the amount of the minerals that are being used by the what? Used up in the soil. Because you remember, you look at plants, and generally these minerals, they are what micronutrients. And these micronutrients are required in small quantity by both humans or by both animals and also plants, because there is also plant nutrition. So when you look at plant nutrition, plants use minerals also in their metabolic activities for different purposes, okay, different purposes. So, and you should expect that the amount of the minerals in the soil, as the plant is using it, it should be depleted. There are also other sources. Also, if you look at the cycle of the nutrient, the cycle also needs to be provided with the exact substrate for the cycle to continue, and that is why fertilizers are being used in so many places to enhance the provision of these minerals, elements that are required by the plants. So also coenzymes, which are small organic molecules that accept and transfer electrons from different enzymatic reactions. They bind loosely to the enzyme at the active site, and it helps catalyze reaction. Usually vitamins and their derivatives from nucleotides examples of coenzyme. For example, we have coenzyme NAD, nicotinamide adenine dinucleotide, which shuttles electrons in glycolysis. So we have the APO enzyme, which is the protein portion and is inactive, and you have your coenzyme, which is a cofactor, non-protein function. That is used as activator. When it binds, it now forms the whole enzyme, which is the active, which is the holoenzyme. So holoenzyme is APO enzyme plus the activator, which is the coenzyme or the cofactor, as the case may be. So if it requires a organic molecule to bind to it, that is a coenzyme. Okay. So when that binding happens, it have now activates the enzyme, and now substrate can come and bind to it, and the catalysis occur. So APO enzyme is an inactive enzyme that requires a cofactor or coenzyme, and the holoenzyme is a complete and catalytically active enzyme because it has its cofactor or coenzyme bound to it. So there are factors that affect enzyme activity. So when you have an activated enzyme or an active enzyme that is ready for catalyzing reaction, you will expect that several factors can either change as increase the activity or decrease the activity of this enzyme. And one of the factors is pH, that is the alkalinity or acidity of the medium where the reaction occurs. So because enzyme functions within a certain pH range, and a change in pH can alter the three-dimensional structure of the protein, and beyond critical level outside its optimal pH range, the enzyme can be denatured. So, and for each enzyme, they have optimum pH or optimal pH. That is the pH value at which this enzyme is having the highest activity. So it depends on where that enzyme is located. That is how you will see the pH value, or pH optima of that enzyme. If it is an enzyme that is active at acidic pH, you see that the pH optima will be within acidic pH. If it is an enzyme that is active in alkaline pH, the pH optima will be in alkaline pH. So outside this range, the enzyme activity drastically decrease, and if it is beyond that critical level, it can also be denatured, and it's going to lose its three-dimensional structure, the quaternary, the tertiary structure, and then get denatured. So example, we compare enzymes that are present in the mouth, enzymes that are present in the small intestine and the stomach. So there is an effect of changing pH on enzyme activity, and also you have the rate of the enzyme activity. With changes in the pH activity, pH on the enzyme activity, you should expect the rate of the enzyme activity to also change. So we have salivary amylase, which is found in the mouth saliva. It breaks down starch to maltose in the mouth, and it has an optimal pH between 6.7 to 7. So it means you can have an enzyme that, if you look at the peak, this peak, look at this blue peak. This is an enzyme that actually have, look at the pH where you have the highest activity. Okay, pH where you have the highest activity is below 5. Okay, and for this, you can have you can look at the pH value. It's the highest peak is at 7. Okay, so it's the salivary amylase that we can see here that has the activity at the highest activity at pH between 6.7 to the neutral pH, which is 7. And this is another enzyme here in green that has the highest peak at what? pH somewhere around 8.5, right? Somewhere around halfway, 8.5. That's the optimum pH for this particular enzyme. So also temperature also affects the enzyme activity. So because they are proteinous in nature, enzymes function within a certain range of temperature. Most of the humans' enzymes, they have optimal temperature around 36 degrees Celsius. Again, beyond a critical level, enzymes can be denatured. So higher temperature for an enzyme that is not requiring high temperature, for example, the human enzymes that work best at 36 degrees Celsius. This is around the normal body temperature. So one thing you should know that enzymes function best at a body condition of the organisms to which they are found. You understand? So for the body temperature of humans, the temperature is normally around 36 degrees Celsius. So most of our enzymes function best at that temperature. There are other microorganisms that are found in very extremely harsh condition. Some can be found in very hot environment, and their enzymes, because of that hot environment, do not get the nature. They do not get the nature because that is the organisms where they are found in, and they catalyze the reaction in that organism. For example, the famous enzyme that we can give example is the enzyme that is used in PCR reaction, which is the Taq polymerase. It is gotten from Thermus aquaticus, which is a bacteria that survives in extremely high temperature. That is why that enzyme can withstand high temperature. In fact, it is active and optimally at that higher temperature. That is why it is being harnessed to be used in PCR reaction. So you can think of the effect of a fever on enzymes in the body. So what do you think now when there's fever, elevated level beyond the normal body temperature? What will happen to the enzymes that are found in the body? So look at effect of changing temperature on enzyme activity, and you have temperature, the x-axis, and the rate of enzyme activity on the y-axis. So you look at the temperature is from 0, the activity is almost, it's almost, you look at it, it's very low. And as the temperature is increased, the activity also is increasing. At 20 is elevated. At 30 is also increasing. And somewhere around 30 to 40, which is around 36, I guess this should be the enzyme of humans. At around 36 degrees Celsius is the optimum temperature. So beyond that 36, the activity is declining. Look at it. Declining at around 40 is coming down, 41, 42, up to 50, it gets down to 0. It means at 50, the enzyme is completely denatured, and three-dimensional structure is being destructed. Okay? So, okay, we have this A and B, and we have a functional protein, and we have a denatured or destroyed protein. You have A, okay, because this is your egg, okay, and when you hit it, what is happening? It gets what? Denatured. It gets denatured because of what? Increase in temperature. And the temperature now acts on the three-dimensional structure of the protein, and it's what? It gets denatured. Okay, this is your this is egg. This is egg. A and B, it gets denatured in B after heating. Okay. So if you look at it here, also you have it foamy, and here soft peaks, and here also you have stiff peaks and overwhelmed. So that is how change in certain conditions affect the activity of your protein or your enzymes. So we have inhibitors, which are molecules that bind to enzymes and they inhibit the true substrate from binding. We have competitive inhibitors, which bind to the active site without being acted on. So they are used in reducing the reaction rate of the true substrates. In other cases, the competing molecule is acted by the enzyme, but again inhibits the reaction with natural substrates. So we have a competitive inhibitor that blocks substrate binding at the binding site. So we have the active enzyme and the binding site, okay, and you have a competitive inhibitor that now comes and block the substrate from binding, and you have what? Inactivate the enzyme. So an interesting example of competitive inhibitors, we have what we call ethylene glycol. Ethylene glycol, which is used as antifreeze in car radiator. Yeah. It's actually a poison which can kill people if it is ingested. This is what is happening. You have alcohol dehydrogenase that is responsible for metabolizing ethanol or alcohol in the body, and the ethylene glycol, which is now, it looks like alcohol and gets converted to oxalic acid, which is toxic by the alcohol dehydrogenase. So how, and this is oxalic acid is very toxic and can kill people. So what are the best treatment ways for ethylene glycol poisoning? We can use an antidote, okay? An antidote can be used to actually block the activity of this alcohol dehydrogenase so that it will not convert the ethylene glycol to oxalic acid. I think one of which is fomepizole or antizole. It's an example of that antidote that is used as alcohol dehydrogenase inhibitor. So we also have non-competitive inhibitors that bind. For the non-competitive inhibitor, they bind at some sites, not the active site. For competitive inhibitors, they bind at the active site. For non-competitive, they bind at other site than the active. They bind at the site other than the what? Active site. They do not affect enzyme substrate binding, but inhibit the enzyme from catalyzing the reaction. Some they act by binding to the cofactors of the enzyme. So we have allosteric inhibition. This is the enzyme and the binding site. You have the substrate that are supposed to bind to the active site, and you have an inhibitor that binds somewhere not in the active site. And the enzyme without modulator is active. Okay, so now when it binds, it prevents, it changes, it actually causes the change of in conformation of the enzyme active site and as a result of that, the substrate will not be able to bind, and you have what the modulator that binds to the enzyme away from the binding side, decrease the what enzyme substrate binding. You have the enzymes being inactivated. We have allosteric modulators which they bind away from the active site, but in doing so, they alter the shape of the active site. This can increase or decrease the enzyme affinity for substrate. We have covalent modulators that binds covalently to the enzyme somewhere away from the active site. It causes change in shape and also the formation and the function of the enzyme. So example, we have phosphate groups, which are one of the most common and important covalent modulators in the human body. You have the active side of the active enzyme. Okay, when now a phosphate group binds to it, it changes the conformation of the active site. So, and we have enzymes that are used in the control of metabolic pathway. Now, this is normally this schematic flow of the enzyme reaction. You have the substrate. This is a metabolic pathway, involving many enzymes in the catalysis of a certain product, a certain substrate to a particular end product. So you have a substrate being acted upon by enzyme 1 to form B, and B acted upon by enzyme 2 to form C, C acted upon by enzyme 3 to form D, which is the what end products, and B and C are the what intermediates that are found, or the metabolites that are what formed during the reaction in the pathway. So what happens when you now control the enzyme? Okay, when you now have what we call the end product now come and act on the first enzyme, okay? And when it acts on the first enzyme, it inhibits the first enzyme from catalyzing A to B, thereby preventing or shutting down the pathway, because the substrate or the intermediates are not provided for the subsequent enzyme to act on. So allosteric inhibition, substrate bind away from the active site and changing the shape and thereby function of the enzyme. In this case above, this can be called what? End product inhibition. It is the product of that particular pathway that now comes and acts on the first enzyme, thereby shutting the pathway. Maybe the body is trying to control the amount of the product that pathway is producing. Okay, so that is how the body tries to use enzyme to control the pathway. So enzyme still on the control of use of enzymes control metabolic pathway. You have your substrate, which is the initial reactant, acted upon by the first enzyme to form B, acted upon by the second enzyme to form C, which are the intermediates within the pathway. And also you can have the third enzyme to be acted upon by the intermediate to form the end product, which is D, or that intermediate can be used by another pathway so that another enzyme E can convert it to E4, can convert it to an intermediate E, and then the final enzyme will now convert it to an F. So this final product may be used as what substrate that can come and now inhibit the first enzyme. This is what we call the end product inhibition. Another example, end product inhibition. The first example, the reactants flow to a particular end product, but in this second example, you have the reactants going to form a particular product, but the intermediates that are formed during the reaction in that pathway can also be taken to form another product. Now that product, when formed, can now come and inhibit the first enzyme. So the inhibiting of the one first enzyme can stop multiple pathways from continuing. So we have enzyme and substrate concentration. They also affect the rate of the chemical of the enzyme reaction. And the rate of enzymatically catalyzed reactions assessed by can be assessed by measuring the products that are synthesized or the amount of the substrate that is being consumed in the reaction. And the rate of the reaction is the amount of product that is formed per unit time, or the amount of the substrate that is being consumed or used up in the reaction per unit time. So it is directly related to the amount of the enzyme present. If the substrate concentration is kept constant, then more enzyme that is present, the more the enzyme that is present, the greater the rate of the reaction. That is, the more product that is what produced. So when you have a specific, or you keep substrate concentration constant, and you have more enzymes, it means more enzyme molecules need the product. So there is going to be what more product, more enzyme is going to be needing the reactants that are present. So that is the greater the reaction that is occurring, and also more product will be what will be produced. Okay, so if you look at the chart here, rate of reaction will increase with the increase in enzyme concentration, provided that the substrate concentration is kept constant. So also, the rate of reaction is related to the amount of substrate present and can reach a maximum point if the enzyme concentration is now kept constant. That is, the enzyme concentration is kept constant, and the reaction rate will increase as the substrate increases, but there is limit to how fast the reaction can go. So when you keep the enzyme concentration steady, and you have your reaction rate can be increasing with increase in the substrate, and it will reach a maximum saturation point where all the enzymes are engaged by the reactants, because the reactants are more and the enzyme is constant, and you have this maximum rate of saturation that occurs. So I think that's where we're going to stop. Thank you for watching. If you are yet to subscribe to the channel, kindly click the subscription button and hit on the notification bell so that our future content will be sent to you. Okay, if you have any question, feel free to comment, to share in the group and also in the comment section. And you can share this video if you find value, so that other people can benefit. Thank you. See you next.